Hi, hello, and other such phrases. Welcome to Microbiology Journal Club.
Hi there. My name is Danny, and in a previous life, I dropped out for my PhD in microbiology at the University of Chicago, where I was infecting skin I grew with MRSA. Nowadays, I'm a fact checker for pharmaceutical advertisers and the president of Biotech Without Borders, a nonprofit based in NYC dedicated to enabling the public with the tools of biotech.
My name is Faz, and in a previous life, I got my PhD in microbiology from Imperial College London, where, amongst other things, I was making flesh-eating bacteria glow in the dark. But since then, I've worked in research integrity, and these days, I'm an editor for an academic journal.
So every week we meet to talk about microbiology, and today is our deep dive week where we look at each figure in a paper that we chose last week to learn and criticize the scientific findings. This is a journal club, so we encourage our audience to leave us questions and
Next week
comments.
will be our news week, so where we survey a bunch of articles and choose which article for the next deep dive. So make sure to subscribe, like, and so you can pay attention and see what we're talking about to satisfy your microbiology curiosities.
Yep, and if you want to see the papers that we cover, they're in our shared Zotero library, linked in the doobly-doo
And
below.
we want to hear from you, so please use the comments or even tweet at us via our various Twitter accounts that are linked kind of underneath our little screens.
All right, so this week we've returned to SARS-CoV-2 after somewhat of a hiatus, looking at other things, because, it's still like a huge microbiology thing going on in the world today.
Yeah, and today we're going to look at a paper titled Vaccination Boosts Protective Responses and Counters SARS-CoV-2 Induced Pathogenic Memory B Cells.
Hmm. And so the first slide that we have up here is sort of the efficacy from the Pfizer vaccine. And so like there are a bunch of vaccines out. I hope people are aware that if you're able to get your vaccine, it should be possible in many different localities. There are many different types of vaccines that are out there. Today, this particular paper is focusing on the mRNA vaccines, and so one of them is the Pfizer one. And it's been shown to have really good efficacy in terms of reducing the amount of severe disease. And that's true both from the phase 3 trials, but also like it's been given out like worldwide. So they even have data that's come in from like whole countries in the case on the right hand side.
Yeah, and we've covered this in like number of our news shows, and also we went through the whole papers looking through the phase 3 trial, and this is one of the big figures from that paper showing exactly the amount of differences in infections in people without the vaccine and people who got the vaccine. And
Yeah.
yeah, it's a pretty big difference.
So it's not 100%. I mean, nothing really
Yeah,
in the world is 100%, right? But it's like a huge difference, right? I think when the phase 3 they were claiming something like 95% efficacious, right, in reducing serious disease after that second dose, and they don't seem to have the percentages on the in this huge set, but it's also up. It's like well above 70.
yeah, it's pretty impressive. The point is that it really helps out. Um,
Hmm.
and in our next slide, we're going to like go into like how this actually happens. How does it work? Um,
Yeah. And I think we've spoke about this before. Like when people make vaccines, right? Like they don't always know like these exact, right, the exact molecular mechanistic steps. But we've been studying immunology for years, and we have this concept of immunology that's quite robust, right, through many different experiments. And it's through that knowledge in that we infer like this is how the vaccine's probably going to work, right? Um,
right. So in this case, the this mRNA vaccine just tricks your cells into making the spike protein. So it almost simulates what would happen in a real SARS-CoV-2 infection, where in real SARS-CoV-2 infected cells you'd see like spike on the surface. In this case, it's got spike on the surface, but it's not really SARS-CoV-2, but it still promotes that same reaction. And
right.
yeah,
And I think in our understanding of, right, like what does it mean that one of the cells in your body are tricked into making this SARS-CoV-2 antigen? I mean, based on this is I pulled this from like Pfizer's. I think it's a deck that they presented to the
mm,
FDA or something like that, like some group in the FDA. And they're like, this is the proposed mechanism. Like these lipid nanoparticles, right, that the mRNA vaccines are made with, they're essentially they fuse to cell membranes, right? And so you inject into your body, and they go and they fuse to a bunch of different cells. you don't really know what cells they fuse to. I mean, maybe it is known, but I wasn't able to dig up those details. In this model, they propose that it fuses to these antigen
Mm.
presenting cells, or APCs. And so those cells, I think we traditionally think of them as like dendritic cells. I think macrophages could also be APCs in some
The-
circumstances. Um,
yeah.
and they, anyway,
Oh
oh,
no,
sorry,
yeah,
go
yeah.
ahead.
They sit in your lymph nodes. So let's say if you got your vaccine in your arm, then the idea would be that okay, some of it will go into muscle cells, but others would end up in your lymph, and your lymph will just drain out into your lymph nodes. And in your lymph nodes, you've got these antigen-presenting cells who are just waiting there, hoovering up whatever like kind of particles there are to present to the immune system to give like a sample of what's floating
Yeah.
around in the body.
Yeah, so they present things like this is foreign. I think that that's like one of the thoughts, right? Like it's like, what is this strange thing that's floating around your body? Maybe we should be responding to it, right? There's a lot of it. It seems like we should be doing something, and so that's the idea that like you get vaccinated, these antigen presenting cells make the spike protein. They show it to B cells and T cells, right, which are like part of the effector arms, I guess, of the immune system. And if those B cells and T cells are stimulated in the right way, maybe they get some other signals, right, that say like there is a dangerous thing going on, right, in the body at this time. They will start growing. The ones that recognize that antigen will start growing, and you'll get more of them. And yeah, and that's the thought,
Yeah,
right? Like if those subsets of B and T cells that recognize the antigen start growing, now you have a group of cells that can enact immunity on that target antigen.
yeah. Uh, so yeah, so this basically shows more data on showing that how the vaccine does induce that adaptive immunity from so
So these are like the early indications that, or like I guess the data that we did have like going into the phase 3 trials. Like this is from before the phase threes were published, right before it was out there. You can just look at the amount of antibody that's being made that's specific to the receptor, or you could look at the T
hmm,
cells that are growing, right, that are specific for that. I'm saying receptor binding domain, right, the spike
right.
protein. And in both these cases, these are like early clinical, like these are early, I guess, data points that get collected. And this was before they even knew that it worked against infections, right? Like this is the type of stuff that you just need blood to
Hmm.
test. And of course, it doesn't tell you whether or not the vaccine is actually suppressing infection. But people look at this and it's like, oh, these are the signals that show us that the immune system is doing something, right? And so that's in phase one and phase two, this is the type of data that encouraged investigators to be like, okay, we're ready for phase 3. We want to see how it works against real disease.
Yeah, I mean these are proxies. They're showing that the immune system is reacting to something, and you're getting an immune response, but you need to know whether that immune response is good because you need to make sure that you're training them to hit the right thing. Um,
Yeah.
and this is
And just, oh
oh no,
yeah,
this
go
is what
ahead.
this
Go
is
ahead.
basically showing, and this with this strength they could go into
Mm-hmm.
those phase 3 trials and say, hey, we know this immune system is reacting in the way that we'd hope immune system react, but is that actually any good? Is our understanding of the disease good enough to show that this is a good proxy for how well our body will fight off the disease?
Absolutely. And I guess the other thing to remember is that then when you go into phase 3, then the only thing they're looking for is like, does it actually prevent
Hmm.
disease? Right? But you can see from these two like snapshots of like the picture of immunology, you can't answer like questions like, does the antigen presenting cell pick up this antigen, right? Like, do the B cells become this specific type of B cell, or do the T cells become this specific type? Like, we aren't asking those questions, right? Because like we're just trying to get the vaccine out there, right, to make things go, right, to give this option to people. And so, yeah, I guess we're in a new era now because that vaccine is out there.
Yeah, I mean, what do you want from us? We had a year. They had a year to do this. I mean,
Yeah, I'm not criticizing them. I'm just
yeah,
like trying to give this
but
perspective because
yeah,
I think that yeah, people get this it's so I feel like I feel this way a lot about science, like having done science at the bench, right? It's like you learn like these very limited snapshots of
Mm,
stuff, but then you like read it in the news and it's like, oh, it's doing this, and there's all these missing questions
mm,
that are yet to be followed up on. In some ways, they aren't important if you just want to get like the end result, but that understanding could help like figure things out that you didn't know before, right? Like it's kind of like you want to get a bigger picture so you can preempt problems that might occur or even answer questions you didn't even know you had.
yeah. Okay. And so yeah, and also like this is picking up on a little bit of our history in this because we a couple of so in the doobly-doo I've linked to one of our previous like talks where we talked a lot about this
Hmm.
paper called I think BCL6 well it's to do with how germinal centers, the place where cells train up to become better at fighting viruses. Uh,
Yeah, I have in front of me. It's loss of BCL6 expressing T follicular helper cells and germinal centers in COVID-19.
Yeah. Uh, so I think the main thrust of that paper was that they looked at people who were like had died of COVID or had really severe COVID, and they found that that
Hmm.
in people with really severe cases, the germinal centers were kind of messed up. Though they're not all the organization that would usually go into cells being going through like hypermutation to become better at fighting disease. That wasn't happening in the way that would usually happen, and they linked
Hmm.
that to the creation of these double negative memory B cells.
Hmm. Yeah, I think that this is actually a very interesting like thought, right? That maybe people have had, right? Like, oh, we already make antibodies towards SARS-CoV-2 when we get infected, right? Like, what does it matter that a vaccine also lets us make antibodies towards SARS-CoV-2, right? Like why is one considered protective and the other one maybe not so much? And I think what was really great about reading this paper was it sort of gave us this potential hypothesis as to why that could be the case, right? Because when you get infected by SARS-CoV-2, right, it's also doing other things to your immune system. And yeah, these double negative memory B cells, which have been linked previously to like improper immune responses, like autoimmunity, and also like an increase during aging, which is also known to be like a time where your immune system is changing. Um, yeah, they had this nice like mechanistic line of reasoning that gives rise to these types of explanations as to why regular SARS-CoV-2 infection wasn't going to be as protective as like another way of getting those antibodies.
Right. Uh, completely. And so the way that it was explained was that the SARS-CoV-2 infection kind of impairs the B cells transition to memory because of that germinal
Mm.
cell center structure. That organization is so important to the development of immune response. And and in these like autopsy patients, they found that germinal centers were all very disorganized. So it's usually you'd have like rows or lines of like cells and like similar cells would be like together and they'd like kind of be interacting with each other in an organized way. And in SARS-CoV-2, they're paired up randomly and kind of disorganized. It felt more like so where you say so I'm going to go back to like an example I kind of came up with in this podcast that I was selling to the do. If you don't have
Mhm.
2 hours to spare to read about the previous paper, we've got a 15-minute explanation of the paper as well. Um,
Yeah, I thought it was a good summary. We kind of helped summarize it a
yeah,
little.
so you want to like in a training dojo, you've got like people practicing moves and stuff, and this kind of turned it into more like a brawl of people just attacking randomly and not no organization. Um,
Mhm.
yeah, so
Yeah, yeah. Like I think the other thing that we didn't I don't think in those episodes we really talked about like that there's the germinal center maturation of antibodies, but there's also like some maturation that happens before
mm,
then as well. And so like that's why I picked out this figure from a review is just to because like we're going to be talking about B cells in this paper, and so like we need some like background, I guess, to get at that. And one of the things that so you can see the starfish like thing that's the antigen presenting cell, right? And so it comes into play twice, right? Like at first antigen presenting cells can engage B cells, right? And then T cells can engage those B cells, and then T cells can start asking B cells to proliferate, right? Let's make more of you. But then, as you were saying, there's this other stage in the germinal center, right, which is essentially the training dojo, which was I think it was a good analogy. I like it. And in that dojo, they actually they mutate
Yep.
and they get selected. So there's like the antibodies on the surface. B cells have antibodies on their surface. That's the B cell receptor is an antibody
Mm.
essentially. Um, and so though they actually undergo mutation, and then only the best, right, are selected again, right, to then continue to proliferate. And in that training dojo, that's also where you get the majority of memory coming out of. Like you get memory out of both sections. So this is like the immune system's
Mm.
crazy, right? Like there's all these checks and balances and stuff, but you do get some memory coming out of that second cycle as well. And so if we, harken back to that paper that we covered, right? You could imagine for people who know immunology, they'd say, oh, well, because you don't have germinal centers, your memory is going to get like messed up in some way. And if your memory's messed up, then you're not highly tuned to making antibodies specific for this antigen, and you're kind of just like throwing it
Mm.
everywhere. Um, and then the one other thing I want to say before we leave this slide is there's this like long-lived plasma cell they also talk about, and we'll talk about it in here as well. Also a B cell, right? So B cells like they can differentiate into a bunch of different forms, and I think the idea is that these are the ones that are antibody
Mm.
factories, right? So like once B cells are stimulated in a certain way, you could have some that stick around and kind of hold the memory for later, but some also just go into full swing and start making antibodies, right? And cells aren't immortal. Well, most cells aren't
Mm.
immortal, and so like they do end up perishing over time, right? And that can be like the decline of immunity. But the idea is that the memory is supposed to stick around so that the next time you encounter something, it can be activated.
Right. Yeah. So now talking a bit more about double negative memory cells, which have IgD negative. So they are sorry,
IgE.
IgD CD27 negative B cells.
Yeah. So IgD, that's the immunoglobulins. That is a type of
Mm.
antibody, you could say, right? It's actually, I think, the most naive form, right? Like IgD and IgM are like sort of the initial types that B cells have. And then they actually, during this process of like getting trained up and growing, they switch over to things like IgG and IgA, which are, I guess, more the typical circulating antibodies that you would get.
Yeah, and they behave.
So memory cells don't. Oh
Sorry.
yeah, memory cells don't have IgD. I guess that's like one of their
Yes,
hallmarks.
that's right. Yeah, and they do respond differently. Uh, I
Yep.
mean, so I've seen them being as so in like HIV, they're associated with being like very inactive or like anergic, but in like lupus, they're also associated with autoimmunity. So there's some odd things going on there. They're often seen at the scene of the crime on various like kind of diseases.
Yeah, and in this, I think maybe they were first described in aging. I'm not sure. I just grabbed
Yeah.
this paper because it was linked and it seemed like a good review of it. But they showed that in young and old people that there's a difference in the amount of these cells, and it's known that as you get older, your immune system sort of ages and maybe isn't able to keep up with some of the things. And yeah, this is just some of that evidence that they showed. So the memory cells, they make IgM, they make IgA, they make IgG. They have the hallmarks of being mutated and going through that process, but they don't really behave the same way. So they are memory. It's just that they maybe don't work the same way as regular memory. And again, like this is this all stuff is like you can't say for certain some things, right, about the immune system, but in general, the thought is that having too many of these cells isn't giving you the best immune response.
Yes. And yeah, so uh, now we've got like vaccination happening. So I mean, I've got my first vaccination. Had some vaccinations, I
Yeah.
assume.
I've got my first vaccination. My second one is coming in on Tuesday. Yeah, so like tons of people have this vaccine in them now. So if you draw blood, right, like this is an opportunity for the scientific community to really understand what's going on, right? Like we can start measuring T cells and B cells, right, in vaccinated, unvaccinated people, and people that have been previously infected, right, and vaccinated, or maybe not previously infected and vaccinated. And all of those circumstances are probably different to the immune
Hmm.
system. And this is the time now that the vaccines are everywhere, that scientists have begun to study it.
Yeah, yeah. I mean,
Um,
this could be interesting for lots of different diseases, not just SARS-CoV-2. I mean, because this presents a massive opportunity for understanding how the immune system works. Because SARS-CoV-2 is
hmm,
so prevalent, we've actually got a huge number of people to get information from that we can use
yeah.
to figure out more things for the future.
Yep, and how these vaccines work,
Hmm.
right? Because also, like, how many people, like, before, 2020, how many people had an mRNA vaccine in them,
Right.
or how many people had even adenoviral vaccines in them? That's like, there, we know that a lot of people, we know it's safe because we've tested like the safety part of it, but how do they work exactly? That is like a still a scientific mystery in some ways, and we could benefit from that knowledge outside of this pandemic.
Yes, definitely. And yeah, so that I think that brings us to um.
Uh, one
Ooh,
last thing,
yes, actually, one last thing.
also from this paper. Yeah, actually, what's really interesting too about this is that um, in this one particular publication, they looked at systemic side effects of getting during your
Mm.
second dose. So I don't know if you've heard, but like I've heard in my circles that that second dose, you do feel like a bit more worse for the wear after getting it. And actually, I even heard some anecdotal stuff from some of my friends being like people who had SARS-CoV-2 earlier during the pandemic that then got the vaccine, they also feel really bad after their first dose. And here's a little bit of data, right, to kind of throw that way,
Right.
right, saying that if you had more spike or RBD specific antibodies, those people were more likely to have these second dose side effects.
Right.
And so, like, again, like that's like kind of a mysterious thing, right? Because it's like we say, oh, you have antibodies against the spike protein, but like that really hides the complexity of everything that's going on in the immune system, right? Because you would say, oh, antibodies means you're protected, but in this case, antibodies also means that you're getting some sort of strange side effects, right? From the vaccine, or you're feeling these things. I mean, they're all manageable. None of them are dangerous in this sense. And again, I think we should also remind people that one of the focuses of doing the phase 3 trials was this antibody mediated disease enhancement,
Mm.
right? That they specifically looked for, right? Cases where it would make the disease worse. It doesn't, right? That was shown. But again, going back to just like how does it all come together? What does it mean that having antibodies ahead of time, right, leads to different outcomes? And so, yeah. Anyways, that's all I want to say here. I think there's like a whole bunch of interesting things that we're gonna be able to learn about, not just how the vaccines work, but also maybe how the disease process goes.
Yeah. So now we're starting off with our population. So this is interesting. So this is conducted at Rutgers University, I believe, where they took a convenience sample. So 83 people who university employees who had been infected during the first wave of the pandemic. So it is a non-random sample. I think it's what you call a convenience sample, where basically you're in your lab and you go, okay, who do we know that's got this disease, and then you just grab
Yeah.
them. And 81 of them had been confirmed by PCR. Others had been confirmed because had a severe disease and had through X-rays and exposure history, they'd kind of deduced that they probably had SARS-CoV-2. Um, and the
Hmm.
patients in this cohort ranged from mild to moderate symptoms. So we didn't. So most of them were like at level symptoms that didn't need them to be like intubated or taken to hospital. So
Hmm.
and between an age range between 31 and 56. Um,
Yep. And for a subset of those, they were able to take 3 to 6 follow-ups for those people and essentially create like this longitudinal cohort, right? Where they every month for 3 months they had a blood draw, and then after that, every 2 months to 7 months, I think, they had a blood draw. And so this sort of data set is really cool because you're getting blood from people who have been infected, right, over time, and even more interestingly, during this period of time, the vaccine came out.
mm,
And so the authors are like really nicely positioned to show us like over time, right? And before and after certain circumstances, what happens in the immune system. And they're interested in memory B cell responses here
yes.
and antibodies as
Hmm.
well.
Which brings us to our first figure, where they took a kind of a very quick like antibody test. So
Hmm.
this is an ELISA. So how an ELISA work is you got your antigen that an antibody will react to. In this case, I imagine it'd be something to the receptor binding domain, and then
Yep.
you have an antibody that's bind. So if you the antiserum, and if there's any antibodies that recognize this antigen, you just bind to it, and then you have a second antibody that recognizes human antibodies that has like something attached to it that will be a reporter, and if that detects something, it will make in this case it will make the solution more cloudy, and so
Yep.
when a laser is passed,
Or like a 405. That's probably some sort of color,
yeah,
right? That's like
that
yellow, maybe.
yeah. So in that case, yeah, it's looking at the absorbance at a specific like spectrum. So this would be yellow,
Yeah.
and it'll be passing a laser through that to essentially see how much of that yellow light is blocked. And in this case,
Yep.
you can sort of see that there is an upper limit. There's a point where all the yellow light is blocked because it looks like kind of an hourglass
Mhm.
shape where at the top you see there's a big bump of things because there's a maximum amount that something can get absorbed. So this kind of thing, so using OD 400 is quite good for like a straight yes or no. Are there antibodies here? And then at a certain point you go, oh yeah,
Mm.
those people up there, they definitely got lots of antibodies. So much it's broken our test slightly. Um,
Yeah, so all those dots are from, I think, the 22, but also like all of their
yeah,
samples, right, are all thrown in there, right? They all did ELISA, and you can see you get antibodies when you've had SARS-CoV-2, right? Different levels of
yeah,
them. And then they also have a cohort as a control, right? People who came up PCR negative,
right.
right, from this group. And then I think pre-COVID is this same people in this cohort that just never got
Oh, pre
COVID-19 or like before
pre-COVID
that.
is
Oh,
interesting because
go
they just
ahead.
had some serum samples on record that were collected before the COVID outbreak that they tested. So
Mhm.
dipping back into our history to see whether this is like test whether there's any cross reactivity with maybe other coronaviruses we've had, perhaps, or uh,
Gotcha. Yeah, so this is like really a good control for making sure that the ELISA is working,
yeah,
right? That there's no cross reactivity in the ELISA.
uh, right.
Okay.
And sorry, the next one is okay. So this is looking at symptoms associated with like SARS-CoV-2.
Yeah. These people had symptoms,
Yeah,
right? Like we said it already in the breakdown of the cohort, but like people were from mild to severe, although none of the severe group were in the longitudinally followed cohort. And this is just like the symptoms that you would see. These are SARS-CoV-2
yeah,
symptoms.
yeah. So most of them got 80% had a fever, then coughing and wheezing.
Mhm.
So this is yeah, we're kind of familiar with this kind of symptom profile. Um,
Yep.
figure one. Okay. So this is looking at the titers of immunoglobulin over time. So this is looking at
Yeah. Yeah. So now instead of throwing it all into that one
mm,
column, they're going to spread it out across like the entire 7 months that these people right have been sampled, and then the lines connect like the individuals, right?
right.
So an individual has many samples connected by a line. They measure IgG, they measure IgD, okay, oh IgM
Yep.
and IgA, and uh, I guess like they don't really talk about the IgM or IgA trends, but in IgG they say that you can kind of see that there's sort of two different groups, right? One that has a sort of a decay in the
Mm.
antibodies over time, and then some that sustain or even go up a little bit.
Yeah, and that's an interesting thing. And they also like in figure S1b, I think they actually do antibody titers, which is so before when we said oh there is an upper limit, how you can get rid of that upper limit is by diluting it down, and then you can get a closer assessment of what the actual levels of antibodies are. So you can do a lot more
Yeah.
comparisons with that, and that's what they kind of did with B. So we've got these titers now that we can see that the differences between the decayers and the sustainers. So like
Yeah, another way, just like by you can modify, right, like essentially the range, right, that you're looking at to try to find where those antibodies, yeah.
yeah, but it just looks like the decays start off generally at a much higher level than the sustainers,
Mhm.
which I find quite interesting.
Right.
Uh,
Yeah, I mean maybe it just makes sense, right? Like bodies aren't like when you make tons of antibody, it's not going to be like that for the whole time, right? Like it takes energy to do that, and so I would assume that this is part of like a normal immune system function. But again, they don't. This is not correlated against anything,
hmm.
right? Like, they don't talk about how this might be influenced by the vaccines, right?
No,
I guess I didn't really look whether or not like they just excluded the after vaccine samples from this particular cohort,
I
this particular
yeah, I don't
analysis.
think they.
This before
I
and after
think this
vaccine.
is like before the vaccines. They don't really pull out exactly which ones of these did get the vaccine or not. Um, so this is they don't really follow up on it. This is something that I think is a nice little buffet piece that someone could take off and look into a bit deeper what the differences between these decay and sustainer like phenotypes are.
Yeah. Yeah, but they don't really speak to them anymore. Just that they exist. There's variance, right, in the way people respond to yeah,
Right.
disease.
Yeah. I mean the immune system is really complicated.
That we'll just keep saying over and over
Yeah.
again, but it's like, oh, we don't really know how to explain too much more mechanistically what's going on.
Yeah,
It's
it's
complicated.
complicated. We need a really smart scientist to figure this out. No, yeah. Oh,
So they also measure neutralizing titers. So they have an assay where they grow HeLa cells that have ACE2 because we know ACE2 is the receptor, and then they have this SARS-CoV-2 that makes GFP, which is glows. And so when they infect their HeLa cells with the SARS-CoV-2, the cells will glow. But if they add serum, the glowing will go down, and so that you know there's something in that serum that's blocking the infection process.
right. And I think what they find is that after like a certain level of like months, that neutralization titer like, seems to go down. Uh,
Yes.
uh,
Yeah. And the ability to neutralize yeah, the ability to neutralize SARS-CoV-2 is positively correlated with the amount of anti-receptor binding domain IgG you have.
yeah, and the furthest graph is mostly like a calculation graph to show like the relative infection rate. So in green is the like cells that are just
It's how they calculate. It's representative stuff on how they calculate the
yeah
NT50, right? Because like it's like a 50% neutralization or something like that. That's like their cutoff. It gives you one number, so they have to measure the neutralization across like a dilution series.
yeah. So that like kind of red dotted line in the middle, that's a cutoff for the NT50 that we've seen in the y-axis for the other graphs on here. So this basically shows they're working, which is a nice thing to see in any paper.
Mm-hmm. Mm-hmm.
Uh, Now, now this is an interesting because this is like how do they figure out whether RBD really is the thing that is causing the neutralization? In this case, they decide to
Yeah. RBD specific antibodies.
yes,
How do they know RBD specific antibodies are the ones that's causing neutralization? Mm-hmm.
yeah. So they depleted the serum of RBD antibodies. So they'd have like same as we've talked about before. They have ELISA plates with which are covered with the RBD, and any antibody would be like with this specific for RBD will bind to them, and they take off all the rest of the serum and test that serum that has been depleted of RBD to see how well that neutralizes, or how
Yeah.
um
I mean, if for anyone who's been listening to us for a while, like I think this is like we're touching on these like themes of biochemical like analysis, right? Like because this is the same concept as when people or when people imagine decoy receptors,
mm,
right? For as a therapeutic, right? It's like uh, these molecules are like floating around in a liquid and you want to be able to grab them but we don't have tiny tweezers to grab them, we just know what they bind to potentially, right? And we can like adhere the stuff that it binds to onto surfaces and like remove stuff in that way, right? Just by exploiting the affinity for molecules that we know exists in other molecules.
yeah. And of course, they did an extra control. They decided to check whether the N antigen might be affected. So N antigen specific antibodies could they also be explaining why the neutralizing response? So G and H are basically them testing whether their depletion has worked and like whether they've actually depleted the serum of these specific antibodies. And I shows how well those serum can affect the relative infection rate after they're applied.
Yeah. Yeah, so the idea, yeah. So looking at I again, right? Like no antigen depletion, right? That serum still is able to in a relative way, right? Reduce the infectivity by like
Mm.
50%, right? But when you deplete things from the RBD, that relative infection rate is still 100. I mean, there's noise,
Mm.
biological variation here, right? Where some go above and below, but essentially, like it's not unable to reduce that infectivity.
Yeah, and N
Um,
apparently that doesn't change things either. So if you get rid of N, it doesn't. There isn't no reduction in infectivity for SARS-CoV-2. So that kind of
yeah.
adds to that story of RBD being very important. Uh,
Yeah, it gets us like much. It's yeah, it gets us a much clearer view, right? A more definite view that RBD is the RBD specific antibodies,
yes,
right? Are the thing in the serum that are helping reduce or helping achieve neutralization.
yes,
So again, this is all in a well,
yeah,
right? So that's not to say that like that's what's going on in the body, right? This is like the biochemical understanding of these neutralizing antibodies.
yeah. So this first part focuses on the antibody story. This next part is going to move on to the B cell side of things. So we've got a
Hmm.
multicolor flow cytometry panel. Uh, so this is
Yeah, so we've talked about flow cytometry before, so I don't think we have to belabor it. Oh, just that like on each axis, right, you're seeing more or less signal, usually from light of some sort, right, that's bouncing off of other antibodies, tags, right? Like we have labeled antibodies that we use to figure out what's on the surface of these cells.
yeah. So I'm going to go through this, try and go this as quickly as I can. So, A, forward scatter versus side scatter tells you the size of the kind of cells and their granularity, so how much stuff is in them. So white cells tend to have big nuclei and lots of granules of stuff that they use to fight things. So you can isolate them out, and then using the LD stain, so are they alive, are they dead? And also whether they've got B cell antigens or whether they don't. So they, in this case, they filtered out all the dead things and all things that they were sure weren't B cells. So things with CD3, CD4, or CD56 or CD16. And then they moved on, and then they got CD20, CD19. Those are what we think are B cells. From that, they can
Yeah,
isolate them further. So plasmablasts. So plasmablasts have CD27 on them. And also
yep,
when you take,
and CD8,
yeah, so yeah, that's right.
CD38.
CD38.
Yeah.
That's right. Then they move off. Now we're going on to the memory cell kind of compartment. We've got IgD and CD27. So you've got our non-switch memory cells, which are have IgD positive and CD27 positive as well. We've got naive cells, which are IgD negative, but CD27 no IgD positive and CD27 negative. Then you've got switch memory, which
Mm.
are IgD negative and CD27 positive, and then you've got the double negative, where they don't have nothing. Um, and
So I think we should linger on
yeah,
this one. I think this is the best one to linger on because what are these four different
right.
things, right? So like they did this strategy to get us to B cells, and then from that plasmablast, so they removed off the ones that are like the ones that are making anti or already antibody
Mm,
factories, right? And now they want to know like which ones are the memory. So IgD positive, but CD27 negative, right? That's naive.
yes.
So that means they have IgD on their surface. That's, I guess, the base. All B cells before they've been stimulated have this
Yep.
IgD, but they don't have this marker that tells them that people have accepted, I guess, for a long time is the marker of memory, CD27. But like strangely, the double negative memory cells like don't have this marker of memory
Mm,
CD27. Like they're weird. Again, like we talked about in the intro, right? Like they're strange. So they don't have what you would think is the marker, but we've studied them before, and so we know that they are some type of memory, and that's why they get in this bottom quadrant. And then we have switched memory and non-switched memory. So class switching is happening during that training
mm.
process, right? As they're undergoing those mutations, as they're getting selected, right? Like we're selecting, they also switch their classes so that they can make, I guess, a more effective type of antibody. They switch away from M usually and D and into A and G.
So I tend to think, sorry, looking at this, I tend to think of it moving from naive. kind of in a clockwise way. So through the cells are going from naive to non-switched to switched uh, in terms
Yeah.
of development.
And they could go from naive to non-switched to maybe double negative,
Yes.
or maybe they could go naive double negative. We're not really sure, right? That's kind of like I think that's probably a big question. Maybe there's already stuff known. We
Yeah.
don't know. I don't know that answer.
We don't know
Yeah.
where the double negative units are coming from in this kind of bit. I only know the first three quadrants are things that,
Well, they definitely come from
yeah, they
naive,
definitely come from,
right? Like,
yeah,
yeah, but we don't know if they like pass through the non-switched memory or switched memory. Yeah, we're not clear on that particular detail.
yeah. And F shows which proportion of the B cells actually respond to the RBD tetramer. So RBD specific B cells in that in F.
Yeah, I don't think we've ever really talked about tetramers. Like we talked about epitope mapping, where they use like peptides and stuff to stimulate T cells, right? To figure out like what T cells are specific to, right? They put the peptide in, and if the T cell responds to it, then in the presence of some interleukins, they expand. Tetramers are funny because like B cells touch T
Mm-hmm.
cells, right? And so tetramers are like the RBD like in that context. And yeah, it's just a bigger chunk of molecule, right, that has an RBD section embedded in it that B cells recognize. So if they're positive for that, we know that these are specific for RBD.
Right. Uh, and
Okay.
here's the full results
So
of the panel.
yeah, all these different cat phenotypes that they are measuring throughout that panel, and they're able to group their cohort in different ways. So in this particular analysis, they looked the difference between non-infected, those who were early in their infection, so under 2 months, and then those who were later in their infection over 5 months.
Right. Yeah. And so you've got a kind of these are the raw numbers of everything. And yeah, the interesting thing. So my eyes immediately go to the DNM like kind of row, and I'm looking at kind of infected 2 months post, and you've got like 10.4, and then suddenly, like in C, they've doubled in rate the number, which is interesting.
Mm-hmm.
And similar increases are seeing in the RBD double negative, which is also quite interesting. Because again, like since these RBD like kind of double negatives are increasing, I'm thinking back to when he saw the kind of the decliners versus the kind of
Mm.
I
Mm-hmm.
can't remember the name for the other group, the group that
Sustained.
sustainers, yeah, the people who like increase. And I'm thinking like, now how much of that increase is due to these double negative cells that are increasing in number?
Yeah. Could it be them? I mean, in some way, like this is we said it earlier, like it's not a really big sample size that they did,
Yeah,
right? But it's humans, and they have these samples from a long period of time, which makes it a very valuable data
yeah,
set. And at some level, like maybe we can't if we dug into the analysis and we like picked out the ones that were decliners and sustainers, we might see like rough sort of correlations, but they would all sort of just be stuff that we have to follow up on
yeah,
later. Um, but I do, I think we sometimes make the plug for open data, right? Like it would be great to see this data set. Like if you think about what it is, right? It's all these samples in a list. They all have a time and
mm,
a patient associated with them, and then they all have like these cell type frequencies associated with them. And you might not be able to pick out particularly statistically significant, right, like correlations in there, but you could potentially find some that would help clarify a particular model, right? That's happening.
right. Yeah. Now figure 2GH. So these are,
They're essentially into that table.
sorry.
They're essentially zooming into that
Yes.
table in G&H. And they want to point out to us, the readers, that plasmablasts seem to be highest in the early samples, the early infected
Hmm.
samples, the under 2 months, um, higher than when people are not infected, and higher than the samples that come from later in the infection, over 5 months. And that's consistent,
Yeah.
right, with what we understand of long-lived plasma cells, right? Unless you're challenged again, they shouldn't expand.
Yeah. Your body doesn't want to waste too much energy fighting a disease that isn't there. So again, this also ties into the decline in antibody levels that we've seen in the previous figures as well, because there aren't as many
Mhm.
plasmablasts out there to just make them.
Yep.
Uh,
And then in H, they're looking at the RBD B cells, and those seem very similar between the 2 months and the 5 months, right? So in that category, right, like there's all sorts of things, right? Memories in there, long lives in there, right? They're all responding. And I guess importantly, you don't see any of those specific RBD cells in the uninfected group.
right. And now figure 2I. So now they looked at the double negative memory cells. Uh, um, so yeah, again, this is zooming in, and again, we're seeing this big like kind of increase in them after 5 months, which is
Yeah.
crazy.
Yeah, so this is the same thing that stood out to you when you were looking at that table. Like the authors also zoom in and show us, like, yeah, that is the case, right? Over time, you get more of this double negative, and it's true both in general, like in all double negative memory cells, but also the ones that are specific for RBD.
Yeah, and I think if we like remember think back to the original paper, the last paper we looked at, these double negative cells didn't want when they're created during infection, not necessarily created to be specific to SARS-CoV-2. They can be there is a generic kind of activation that happens that so just because they aren't like say specific for RBD doesn't mean they weren't triggered by the SARS-CoV-2 infection. So I
Yes. Yes. Yeah, like it could have been through that mechanism. Like this is the great thing about I think that we had read that paper
yes
beforehand and then we got to read this is that we can see that that's like a hypothesis that can be drawn, right? Like the triggering of or sorry, the destruction of the germinal centers, right, by infection. Again, we don't know the mechanism of that, but we know that it does
mm,
happen. Um, that could be leading to, right, this increase in double negative memory cells.
yeah uh,
And I think that's also attractive. Like again, if we're in the hypothesis space, like that's also really attractive from the standpoint of double negative memory cells could be have been known to be associated with like certain types of autoreactive
mm,
antibodies, right? And we know that some of these clotting things that happen during infection are because of autoreactive
yeah.
antibodies, or it could be because of autoreactive antibodies. So again, we're like filling in that story, right? None of the solid links have been built, but like this paper is trying to set the groundwork for asking those types of questions.
Yeah, I think it brings shines a light on this specific part of the immune system that we might not have like looked at before because like say SLE, lupus, and HIV they're not necessarily this is not the focus for the work. So it's interesting that now we're looking at it in the context of a disease that is affecting everybody worldwide. I think that'll prompt a lot more work into this subset of B cells.
Yeah.
To Okay, yeah, pre-vaccine and post-vaccine. So now we're at the point where people are starting to get vaccinated in this group. So I think they get about 12 people who. Okay, so 12 people donated blood 2 weeks after the second vaccine dose, um, and they did done it before. So they had pre and post-vaccine samples for these people. And again, this is again it's a small sample size, but it's strengthened by the fact that you've got like multiple samples from the same people. So people are essentially acting as their own controls. So that
Yeah, you can see the trend. I feel confident that we're seeing trends in individuals. Whether or not in the global population, everyone will follow that
hmm,
trend, I don't know, right? But like within the people, like these are definitely what's happening here. Yeah.
yeah, and again, like there's some interesting things to pull out here, but I feel like they do focus in on the interesting stuff via some of the graphs they're going to pull up. So we'll go to some of them.
Yeah, that sounds good.
Just go to the graphs.
So the first thing they want to show us is that the vaccination increases the receptor binding domain specific IgG titers. And so like you can see that, right? Because there is in the vaccine
Right.
plus, right? That's higher than vaccine
Yeah.
minus. uh,
Yeah, it's a bit difficult to see because we're not on like a log graph, but yeah, you can definitely see that being vaccinated gives you a better immunity than being infected. But
Yeah, and you can zoom in. Like they zoomed in B to help us see that,
Yes.
right? Like that's the zoomed in section at below. Yeah, I guess that's if they did log, I guess they wouldn't even have to have done this. Is that what
I
that's
mean,
part of
I'm not
what you're
sure,
saying?
but yeah, you're right. B is where they zoomed in to show that actually getting vaccinated gets you a better immune response than being infected, which is kind of throws, which is great for. I mean, because I think there has been a strong like kind of push for people who think that getting infected is as good as getting vaccinated, but that's
Mm,
kind of a dangerous line of thinking because again, like SARS-CoV-2 is not a fun disease to have. It can cause side effects. Even if you do survive, you can have long-term symptoms. Uh, so we really
hmm
do want to have you become immune in the safest way possible, and that's through vaccines.
Yeah. And then remember, like also from like the paper that we knew before and like the whole all these hypotheses that we've been weaving into
Mm,
this data, right? That like there's this idea that like when you get infected regularly, something else is happening in the immune system that isn't like the ideal immune response.
right. Yeah, and the thing that also is quite interesting, but I guess you'd kind of predict because people who've been infected before who get vaccinated get a much bigger antibody response because uh,
hmm
that's
hmm
again that's going back to our stories about people who've been infected get a first vaccine they have to like sleep for a week or something. Maybe they don't have, but
Yeah. Well, I think it's also, I mean, in some ways, this is this concept is vaccine technology sort of in
mm,
action, right? That everyone should understand. Uh, People are like, why do you get a booster? Why do you get boosters? What are boosters
yeah,
good for, right? Well, they boost the response. You already have some existing memory and response, right? And then it gets boosted by getting vaccinated, right? Because you're seeing that antigen again, and your body's like ramped up to the max. And so this is what we're seeing here, I think, as well, right? Like if you've seen it before, your body knows something about that,
yeah,
right? And that seeing it again in another context, you get even more of a response. And again, we don't really know is that good or is that bad, right? Because like having antibodies, of course, we think it's good, but as I said interestingly in the discussion, right, that there was also some correlation between getting a bunch of antibodies, right, and side effects.
yeah,
So yeah,
yeah, and
you still don't want to be infected before you get your vaccine, but if you do, it's fine. Like it's still safe to get your vaccine. It's just that yeah, you might feel a bit worse for the wear.
right. And now moving to 3CD and E, which is where we look at the B cells. Am I right? Yeah.
And also the neutralizing capability, both
Yes.
neutralizing first is in C and D, so same patterns, right,
Right.
that get more neutralizing antibodies when you get vaccinated, um, more than being just infected by itself, and if you have been infected previously, even more than that.
Right. Yeah. Uh. Oh, yeah. That's. So that's really interesting. And
And then the same pattern with B cells, that if you get more when you get vaccinated, oh
yeah.
yeah, you get more when you get vaccinated, even more so. Actually, this pattern is a bit different. No, it's the
Yeah.
same. Um,
So this is looking at memory B cells, right? And people who get vaccinated still have more memory B cells than people who haven't been vaccinated but have been infected. And people who have both have more memory B cells than the other two.
Yeah. I think this is I like this data. It shows the value of getting the vaccine in my
Yeah.
mind. Like again, like you could draw out the mechanism for a lot more, but like having more data to say that like these are good mechanisms, right? Like for vaccine protection, and we're seeing more of it, right? When you get vaccinated, regardless of whether you've been infected previously or not. I think that's I think it's good.
Yeah. Uh, and I believe that brings us to this is figure S4, which is oh, so drawing our attention to two main things: the plasma blast levels. Um, so
That there's no big difference in the plasma blast levels between between what was it? It's both these cases,
yeah,
A and B. So between infected people pre-vaccination and post-vaccination, right? And non-infected people pre-vaccination and post-vaccination. So you're always getting the same increase, right, of
Right.
plasmablasts. Oh no, no. Sorry, you're not really getting a big increase in these plasmablasts, is what they saw.
Yeah. But and it seems that like so there's some so I mean so tell me about the switch memory cells. So what's so I'm
Yeah. So then the difference. So then there's a difference though in so 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 no difference in plasmablasts, but in switch memory, they're seeing a decrease, right?
yeah,
you get less you get less switched memory cells after being vaccinated.
right. Okay. Compared to being infected, yeah. That's interesting.
It's A and B. It's in both
Hmm.
cases, right? It's in both infected and non-infected. Um, like the vaccination has this effect on you, right? Less switched memory, same amount of plasmablasts.
Right. Oh yeah, I see. Yeah, I see what you're
And
saying there.
yeah,
Yeah.
I so the significance of this, I know. I mean, these are just um, I guess it's showing like how the vaccine
Hmm.
works. So the vaccine works regardless. Well, first of all, it seems like this is quite similar between um yeah, like this is similar between whether you've been infected before or not
Yeah.
infected before, right? Vaccine is having the same effect on in these measures of the immune system. That's one thing that I'm getting
Hmm.
from this. And then the second thing, I guess, is maybe something about how the vaccine is working, right? That it somehow like you don't need it. Maybe it's like not about the frequency of the switched memory cells, right? That matters, right? Like, maybe you know what I would have liked to see. I think would have helped to interpret this a little bit better. Let's overlay this with RBD
Right.
specific.
Because I mean the
Yeah, it might.
yeah. Because I was
Mhm.
thinking that I mean there's some ways you can interpret this depending on because if switch memory cells I mean yeah if you because these are from blood samples, so
Mhm.
I mean we could interpret this as saying that the vaccination is reducing the number of switched memory cells, or you could say that the
Mhm.
switch memory cells are all in a different place that aren't so they're not circulating the blood as much because they're busy in the germinal centers or potentially. I don't know whether that makes sense
Oh
or
yeah,
not.
I don't know. Yeah. I think that's like that's definitely out of my realm of immunology
Yeah, I
expertise.
don't because
Um,
yeah, I mean I don't think that makes sense because again the way I thought about it was that switch memory cells are being created in the germinal centers and then filtering out in the blood. I so I don't really yeah again my knowledge of immunology is hitting a wall for this point. Um,
yeah, in terms of how to interpret it,
yeah,
right? Like they've shown us these differences, but what does that mean, really? Right? And so, yeah, the way I summarize, the only thing I'm taking away from it is that the vaccine works similarly between infected and non-infected people, and that it does have some effect that, in my mind, is actually counterintuitive. I would think there would be more switch memory, but they don't overlay RBD specific on here.
right,
So, like, that may be part of the story that we're missing, right? Like, it maybe RBD switch memory specific does go up, right? And it's other ones that are going down. We don't know.
yeah.
We can't know from this.
But I think the thing that we're focusing on a lot more is the double negative memory cells. Because yeah, with vaccination status, that it changes the numbers of them. Um, yeah.
Changes the numbers of them for those who have been previously infected.
Yes, that's right.
Right? So this is so in one place there is a difference in how the vaccine affects you, and that is in your double negative memory cells, based on their analysis.
Right.
And that sort of makes sense in this sense that people don't have those double negative memory cells unless they've been previously infected.
Yeah, I mean, yeah,
Right?
the ones in the blue dots are definitely like at a low baseline, and then suddenly
Mhm.
the third column, you see this big boost in the number of double memory cells, and then after vaccination, it goes back to baseline.
Yeah. So, I mean, if we interpret this in light of that paper that we covered before, right? Like this is actually giving us like another type of protection that the vaccine is giving us, and that type of protection isn't the type of protection that you would have thought of in terms of like antibody titers, right? Like it seems to be affecting us it affects people similarly. It gives people who are infected more antibody titers, but it also reduces the amount of double negative memory cells, right, that they have. And maybe that's good. Again, it's not correlated against outcomes for any of these circumstances, but it's giving us this insight into the differences in immune responses that are happening from different contexts.
Yeah, and I think that's the paper. I think and it's really interesting actually. Um,
Yeah, no, I mean it's short and
mm,
sweet in that way, right? It draws our attention. It extends the story of double negative memory cells. I think that that's one of the strong things that we can say that this paper does. Extends that story to the status of
right.
vaccines, right? And like how they interact. There are many mechanistic questions about how these cells arise and what role these cells play in disease that this paper doesn't address. Um, but again, it does actually, it gives us information that we had not seen before. And for me, I was saying that in the introduction, they were saying like there's a huge influx of papers like this
Mm,
now that are coming out because people have been vaccinated, right? I tried to nod to that
yeah.
in the introduction. Because people are being vaccinated, we're getting information like this because you have blood samples from pre-vaccination and post-vaccination, right? And we know that the vaccine's working, but how it works, that's where scientists, I think, are spending their time now.
Yeah, and I mean this shows a really interesting way that vaccines could potentially be benefiting us. But again, there's so many questions about what these double negative memory cells are doing, and what is the vaccine doing that reduces their number? How does that work? And we don't really know at the moment. Um, oh
Uh, what do you think about they in the discussion that the authors put out? They say some stuff about long COVID, what it might say for long
yeah,
COVID. Uh,
because I'm very careful about that because this, I mean, firstly, would what I'd really like to see have seen through them is potentially were there any sequelae? So whether in their infected cohort, do they have people who were showing symptoms of long COVID, and whether those had changed or not? And I would like to have seen because if they did have them and then shown that people those symptoms had been reduced, then that would be a lot more stronger kind of justification for them to say that.
Sure. I mean, I think that there is, like, again, a very great data set,
Hmm.
right, that allows us to ask questions that we've seen in front of us. But there is a weakness to the data set in that they don't collect, it doesn't seem like they collected the symptoms with every blood. Like, it's
Hmm.
unclear what they collected at each study visit, right? I know they collected blood and they showed us a table of symptoms they collected.
Yeah.
It would be interesting to see if those symptoms overlaid on the samples also gave us another story that maybe gave it more of that. Significance in the clinical sense, right? Like clinical outcome significance.
Yeah, and the thing about like talking about long COVID is it's a big like nebulous disease that we don't necessarily understand fully. And this paper,
Mhm.
like in the discussion, they provide a very simple explanation for it, which is very attractive for me because I'm like, oh yeah, that makes sense. But then we need to be very careful when you have a simple explanation like that. Uh,
Yeah. What did they say exactly? I'm not
I
sure if I
think
recall.
I don't know exactly what they say because I don't think they outright say like that it prevents long COVID. I think they mostly say that that could potentially explain the decline in the sequelae, especially in people who have had like long COVID and had a vaccine and then had the symptoms go away. Um, so that's
Yeah.
something that's been
Oh yeah.
observed.
The still anecdotal reports of symptomatic relief after vaccination in persons with post-acute sequelae of SARS-CoV-2 infection, they give two references, may find mechanistic grounds in these findings.
Yeah, I mean, again, I mean, they even say they're anecdotal reports. So even the authors of this paper are knowing that they're kind of pushing things a little bit by saying that. But I
Mhm.
think it is an interesting hypothesis to explore for the future. But again,
Mhm.
I'd want to be very careful about putting a simple explanation on something that could be like not just one but multiple different disorders. So it's
Yeah. Another thing that I think I learned from this paper is just fleshing out the differences between, or like fleshing out the fact that we can see antibody responses. That doesn't always mean we know what's happening on the immune side, right?
right.
Like there's this other story about other cell types that contribute to things that we can find differences in. So even if like in both cases antibody titers are up,
Mm.
or antibody titers are up more in this one, and it's also similarly increased in this other circumstance. There's still something underlying that we have to talk about, especially when we're getting into the nitty gritty of like disease processes, right? Because I think we've said it before, and we both have this understanding that disease can be equally contributions to disease can be equal from the host and the pathogen.
Yeah. Yeah. So yeah, I think that we've basically said most of. This has been a nice paper because we've nearly kept it under an hour for the first time for a while. So, uh, this is. It's been.
Yeah. So great time to
Yeah.
wrap up then. Join us next week for our news week, where we're going to survey papers to find something to cover, just like we did this week.
And we want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it's possible we didn't get everything right. Science is about thinking critically and asking the right questions. So if you have any questions or corrections, please let us know in the comments.
I totally agree. You can reach out to us over Twitter with the hashtag #MicroTWJC, and we both believe that peer reviews is a process which we can all participate in. So we hope that you've had a good time listening. And if you do think that you want to participate or add something or find something unclear, just let us know.
Yeah, it's been a pleasure chatting with you, Danny.
Same here, Fuzz.
Tune in next week for us talking about all things great that are small.